Hey, how are you? I'm Isabella Gonzalez and I'm in third semester of medicine, Universidad Pontificia Bolivariana. And today we're talking about how YiaC and CobB regulate lysine lactylation in E Coli. But what is E Coli? It is a bacterial member of Enterobacteria family and it's going to be part of the microbiota of the gastrointestinal tract. This one is a gram-negative bacillus, and the different clones have been described that due to the para-adaptation processes, these have acquired genetic elements or mutations that function as virulence and fitness factors. This one is going to determine the pathogenesis of this bacteria in different diseases. But what is important about E. coli is its genome structure, because this is highly flexible, allowing the mobility of genetic material through transposons, insertion sequences, bacteriophages, and plasmids. What is important to know now is that YiaC functions as a lysine lactylase, while CobB serves as a lysine deslactylase. This is going to be important in the regulation of the metabolism. YiaC catalyzes the addition of K-like, that is lysine lactylation, and that is a PDM, that is a post-traductional modification, while CobB erases this PDM both in vitro and intracellularly. This is going to be important because CobB can especially modulate the formation of PGKF, that is going to catalyze the formation of pyruvate in the last step of glycolysis. This last step of erasing that K-la can enhance glycolysis and promote E. coli growth. The objective of this study was to identify if YiaC and CobB regulate lysine lactylation in E. coli and illustrate the molecular mechanism of K-la-mediated regulation of glycolysis. In this study, they used a lot of methods, but today I'm talking about 4: immunoblood transformation, SDS page, and in vitro assay. In immunoblood, they separate the proteins by electrophoresis and detected with antibodies the antigens of them. This was important because they want to know if there was K-la present on prokaryotes, and they used the Western blot assay and confirmed that cherry. Secondly, they used transformation. That was important to change the genetic information of the bacteria by overexpressing or not expressing any enzyme that helps to regulate K-la. SDS page is going to separate proteins according to their electrophoretic mobility. This is going to be important to analyze K-la peptides because proteins are going to be desnatured and they're going to lose the three-dimensional for conformation. And in vitro assays, they use it because in vitro assays produce exact results. So anything they want to look is actually in the organism. K-la was originally identified as an instant maker in eukaryotic cells. It was not clear if the new PDM existed in prokaryotes, so they performed a Western blot assay confirming the existence of K-la in prokaryotes. They want to identify the regulatory enzymes of K-la to understand their function and the regulatory mechanisms of prokaryotes. So they hypothesized that some CATs and desaltylases can catalyze the addition and removal of K-la. So they employed overexpression of E. coli strains G and AT to detect the lactylase candidate by immunoblooding. And they found that only YiaC overexpression caused an extensive increase of K-la, and YiaC is candidate lactylase that catalyzes the generation of K-la. They also analyzed the overexpression of CobB, where CobB is the major desaltylase known in E. coli, and these results indicate that CobB functions as an endogenous desaltylase, confirming all the hypotheses. In the second result, they got that in E. coli there is a protein that can catalyze lactate to lactato. To confirm it, they detected lactil-CoA in E. coli as a control. As expected, they detected a low level of lactil-CoA, showing that lactil-CoA is present in E. coli. Next, they incubated lactate with fresh whole cell lysates and found that lactate could be effectively converted to lactil-CoA at YiaC. YiaC catalyzed this action with just acetylation but not succinylation. In the final result, we got that YiaC and CobB can regulate the K-la levels of diverse metabolic and biosynthesis enzymes. CobB can especially erase K-la from GPMA, MDH, and PGKF in vitro. It is important to understand that CobB can clear deslactylase, pyruvate kinase, that is PGKF, and MDH and GPMA in vitro. So CobB can widely deslactylase glycolysis enzymes in vitro. In discussion, we have the word of 4 people: Sandy and Lee L, Liu H, and Olive AJ. Sandy and Lee L said that K-la has an extensive regulatory effect on metabolism in bacteria, distinct from the regulatory transcription of K-la in eukaryotes. We have that K-la in eukaryotes is its own creator or maker, while K-la in prokaryotes is going to be part of the regulation of the metabolism in the bacteria, while they can enhance glycolysis and make more bacterial growth. So yes, this is correct. Liu H said that changes in metabolism can mobilize regulatory enzymes to regulate acylation levels such as lactato accumulates in E. coli under glucose fermentation. This is true. It has a lot to go with the article because they take that in glycolysis anaerobic the lactate accumulates and that is going to be part of the lactyl CoA. So yeah, that is also correct. And Olive A.J. said that host microbial metabolites such as lactate as an example, are a critical factor influencing many psychological processes and the development of diseases. This is also true, but in Entericia coli, this metabolism is important for their growth and their enhance of glycolysis. So yeah, that is also true. So these three words from these four people are important to understand that this article has a lot to go and actually is completely correct. For the conclusion, I thought about these two. The first one said by understanding how prokaryotes use lactate as an important metabolite in their proliferation, it will allow us to find better treatment based on enzymatic regulation of the metabolites mentioned in the article. And secondly, we got that the regulation of enzymes such as YiaC and CobB, which have lower enzymatic functions, can open doors in the field of microbiology. This is all for today, and I'm so glad that you can read this article. So thank you. Bye.